Chapter 3, Cells and Tissues. The learning objectives for Chapter 3, there are quite a few. Name and let's start with cells. So they're considered the structural units of living, and the human body contains 50 to 100 trillion. Robert Hooke was looking through a crude microscope at work in the 1600s. He noted the cube-like structures that reminded him of long rows of monks' rooms, otherwise known as cells, and the name just stuck. Since the late 1800s, cell research has remained relevant and fruitful for a number of reasons. It has provided us the four key concepts known collectively as cell theory. So a cell is the basic structural and functional unit of any living organism. So when you define cell properties, you're defining the properties of life. The activity of an organism depends on the collective activities of its cells. According to the principle of complementarity, the activities of cells are dictated by their structure, which determines their function. So structure determines function. So remember, we learned in Chapter 1, anatomy is structure, physiology is function. Continuity of life has a cellular basis. In other words, cells come from cells. So as we move forward, note that there are many different types of cells, but for our purposes, we will talk about the quote-unquote generalized cell. In general, cells have three main regions: the plasma membrane, a nucleus, and the cytoplasm. We will learn more about each in the oncoming slides. Here we have the plasma membrane. It is a flexible, fragile, and transparent barrier that contains the cell contents and separates them from the surrounding environment. It sets the boundaries of the cell, but acts more like a passive envelope or baggie. You can see in the image that the plasma membrane structure consists of two phospholipid layers arranged tail to tail. If you recall, the phosphate head is hydrophilic, which keeps it turned out to the extracellular fluid and turned into the intracellular fluid. So that outside layer is loving that extracellular fluid, and that inside layer, that phosphate head, is loving the intracellular fluid. The fatty acid tails, pictured at the center of the membrane structure, are hydrophobic, which keeps the inner contents of the cell from going out and the outer contents from coming in. Some phospholipids have sugar groups attached to them, forming glycolipids. You can see some scattered cholesterol and proteins throughout the plasma membrane as well. The proteins, which can move about, form a constantly changing pattern, or mosaic, which leads us to what is called the fluid mosaic model. So a really cool function of the plasma membrane is how quickly the cell wall can reorient itself when disrupted. And I want you to start thinking about how, why is it able to do this? And it's something that we'll discuss more in depth in class. The role of proteins in the plasma membrane. So proteins are enzymes. They act as receptors for hormones or other chemical messengers. They can act as the binding site for anchoring the cell to fibers or to other structures inside or outside of the cell. Most are involved in transport. For example, some will cluster together to form protein channels or tiny pores through which water and small water-soluble molecules or ions can move. Carriers, they bind to a substance and move it through the membrane. Glycoproteins. So glycoproteins determine blood type, act as receptors for certain bacteria, viruses, or toxins, and play a role in cell-to-cell recognition and interactions. So a glycoprotein is when branching sugar groups are attached to most of the proteins abutting the extracellular space. The surface is sugar-coated, so it's sticky and fuzzy, and that part is called the glycocalyx. And that's how it helps to act as a receptor and bind to things. Definite changes in glycoproteins can be seen when you're looking at a healthy cell versus a cancer cell. Cell membrane junctions. So let's talk more about the glycoproteins. So the glycocalyx is what I was talking about on the outside, those sugar chains that make it nice and sticky and adhesive. The wavy contours of the membranes of adjacent cells fit together in a tongue and groove fashion, and then special cell membrane junctions are formed. While certain cell types are free-floating or quote-unquote footloose in the body, some cell types are bound together, like epithelial cells, which we'll talk about later in the chapter, and they can be bound together in 3 ways. The image on the right shows cell junctions of the epithelial cell. So this cell in the picture is epithelial. And you can see the 3 junction types, which we'll talk more about. Tight junction, a desmosome, and a gap junction. You can also see the microvilli at the top, and these just increase the overall cell surface area by a lot, and that increases absorption. Let's discuss a little further the cell membrane junctions, the 3 different types. So first we have the tight junctions. These are impermeable. They encircle cells and bind them together like leak-proof sheets. They prevent substances from passing through the extracellular space between cells. An example would be in the small intestines. So these junctions prevent digestive enzymes into seeping into the bloodstream. So they keep things that we don't want into other places out of there. Then desmosomes. You can find these in heart muscle cells and skin cells. They're anchoring junctions scattered along the sides of adjacent cells. They prevent cells subjected to mechanical stress from being pulled apart. Obviously, you don't want your muscle cells that are working very hard to pump blood to your body, pump oxygenated blood to your body, to tear apart. Structurally, these junctions are button-like thickenings of adjacent plasma membranes, also known as plaques, that are connected by fine protein filaments. The thicker protein filaments extend from the plaques inside the cells to the plaques on the cell's opposite sides, and this forms an internal system of strong quote-unquote guy wires. I'm not entirely sure why they call them guy wires. Gap junctions, so function mainly to allow communication, commonly found in the heart and embryonic cells, and their chemical molecules, they allow chemical molecules such as nutrients and ions to pass directly through. So in gap junctions, the neighboring cells are connected by hollow cylinders composed of proteins called connexons that span the entire width of the membrane. Because they span both membranes, these connexons are called transmembrane proteins. So we just talked about the plasma membrane. Let's move on to the nucleus. So the nucleus is the cell's headquarters or control center, and it's where DNA is stored. It's often oval or spherical as pictured here in this generalized animal cell, but it will conform to the general shape of the cell. It has 3 recognizable structures. First, let's talk about the nuclear envelope. So you can see a better image of it on the right. It's that outside double membrane barrier. It is also known as the nuclear membrane. Between the membranes is a fluid-filled space. And you can see at some points in the membrane, it fuses together and creates nuclear pores, those little orange dots. These pores will allow some, but not all, substances to pass through. The nuclear membrane also encloses a jelly-like fluid called nucleoplasm, which just contains other nuclear elements. Next, we have the nucleolus. So a nucleus will contain 1 nucleolus or more nucleoli, dark staining round bodies. These are sites where cell structures called ribosomes are assembled. Most ribosomes will migrate to the cytoplasm, where they serve as sites for protein synthesis, and we will talk more about ribosomes later on in the lecture. Lastly, we have chromatin. When a cell is not dividing, the DNA is carefully wound around proteins called histones to form a loose network of beads on a string called chromatin. When the cell is dividing, the chromatin threads coil and condense to form chromosomes. We will also talk about that process later on in the lecture. Now let's talk about the cytoplasm. So this is the cellular material found outside the nucleus and inside the plasma membrane. It is the site of most cellular activities and made up of 3 compounds: cytosol, which is a semi-transparent fluid that suspends the other elements; inclusions, chemical substance that may or may not be present, so like lipid droplets common in fat cells, glycogen granules abundant in liver and muscle cells, pigments such as melanin in skin, mucus or other secretory products, and various kinds of crystals, often referred to as the cellular pantry. And then organelles literally means little organs. It's the metabolic machinery of the cell and carries out specialized functions for the whole body. Here's where we're going to start diving a little deeper into the overall structure of the generalized cell. So many organelles are bounded by a membrane similar to the plasma membrane. These membrane boundaries allow organelles to maintain an internal environment quite different from that of the surrounding cytosol. This compartmentalization is crucial to their ability to perform their specialized functions for the cell. We will learn more about the different organelles in the upcoming slides. Mitochondria. So I always thought mitochondria looked kind of like a little pill bug. I think a lot of people liken it to a sausage, but they do lengthen and change shape continuously. So the structure is a wall that is equal to 2 plasma membranes, so it's very thick. The outer part of that membrane is smooth, and the inner membrane has shelf-like protrusions called cristae. The function is that enzymes within carry out reactions in which oxygen is used to break down food. What do you think is formed? So as foods are broken down, energy is released. This energy escapes as heat, but some is captured and used to form ATP molecules, which we learned about in the last chapter. They create the most ATP and are considered the powerhouse of the cell. They replicate themselves by pinching themselves in half, and cells that are very active, like liver or muscle cells, have hundreds of mitochondria, and those that are fairly inactive have much fewer. So something like an unfertilized egg that's not, growing or changing will have fewer mitochondria. Ribosomes. So the structure is their tiny bilobed dark bodies made of proteins and one variety of ribosomal RNA. The function, they're the site of protein synthesis in the cell. The free-floating ones manufacture proteins used inside the cell, and others attached to the rough ER, Endoplasmic Reticulum, which we'll talk about here shortly, which manufactures proteins used outside the cell. So you can see in the photo, they're just ribosomes with the little circles pockmarked on the rough ER. So the endoplasmic reticulum, from now on known as the ER, so I don't want to keep saying that word. So the structure is a system of fluid-filled tunnels that coil and twist through the cytoplasm, continuous with a nuclear envelope, and accounts for half of the cell's membranes. So this is where the ribosomes that we just looked at were attached to. So the function is it's a mini circulatory system for the cell. So there's a rough ER and a smooth ER. So the rough ER is the one that is studded with those ribosomes. All building materials of cellular membranes are formed either in it or on it. And the smooth ER communicates with the rough ER, doesn't have any ribosome, so it has no role in protein synthesis. And it functions in lipid metabolism and detoxification of drugs and pesticides. So these would be plentiful somewhere like the liver that is charged in our body with detoxification and metabolism. So it carries substances, primarily proteins, from one part of the cell to another. The rough ER, the proteins made on its ribosomes migrate into the rough ER tunnels where they fold into their functional 3D shapes. And then these proteins are then dispatched to other areas of the cell in small sacs of membrane called transport vesicles, and they carry substances around the cell. Abundant in cells, the ER is abundant in cells that synthesize and secrete proteins like pancreatic cells. So this image is just showing more about that synthesis and export of protein out of the rough ER. So you can see step 1, as the protein is synthesized on the ribosome, which is attached to the outside of the ER, it migrates into the rough ER tunnel system. Step 2, in the tunnel the protein folds into its functional shape. Short sugar chains may be attached to the protein, which we just learned about, glycoproteins. And then step 3, the protein is packaged in a tiny membranous sac called a transport vesicle. And then 4, the transport vesicle buds from the rough ER and travels to the Golgi apparatus, which we'll learn about here shortly, for further processing. Okay, the Golgi apparatus. So you can see it actually looks very similar to the endoplasmic reticulum. So the structure is a stack of flattened membranous sacs that are associated with swarms of tiny vesicles. It's generally found very close to the ER. And the principal function is a traffic director for cellular proteins. It modifies, packages, and ships proteins depending on final destination. So this is like the Amazon warehouse of proteins. You can see in the image that protein-containing transport vesicles pinch off of the rough ER and migrate to fuse with the Golgi apparatus. As they travel through the Golgi apparatus, they are sorted and slightly modified if necessary, and then packaged within secretory vesicles. These vesicles then leave the Golgi apparatus and head to various destinations. So there's 3 pathways shown here. So pathway 1, Golgi vesicle containing proteins to be secreted becomes a secretory vesicle. So you can see it moves to the outside of the cell and secretes that protein to the extracellular fluid. It just expels it completely from the cell. Pathway 2, the Golgi vesicle containing membrane components fuses with the plasma membrane and is incorporated into it. So it becomes part of the plasma membrane. And then pathway 3, the Golgi vesicle containing digestive enzymes becomes a lysosome, which we'll learn about here shortly. And the lysosome fuses with the ingested substances to digest them. So we just heard the word lysosome in pathway 3, but they're basically the breakdown bodies. In structure, they appear in different sizes and are membranous bags containing powerful digestive enzymes. Function, lysosomal enzymes are capable of digesting worn out or unusable cell structures and most foreign substances. Their cellular stomach. So it just takes care of whatever the cell no longer needs and it digests it. It's abundant in white blood cells called phagocytes. So the enzymes they contain are formed by the ribosomes on the rough ER and packed by the Golgi apparatus. Then peroxisomes. The structure is a membranous sac containing powerful oxidase enzymes. They look like small lysosomes, but do not arise from the Golgi apparatus. They replicate by pinching half or bud directly off of the ER. Their function is to detoxify a number of harmful or poisonous substances. They disarm dangerous free radicals by converting them to hydrogen peroxide, and then catalase converts extra hydrogen peroxide into water. They're numerous in liver and kidney cells. So free radicals just being highly reactive chemicals with unpaired electrons that can damage the structure of proteins and nucleic acids, which are normal byproducts of cellular metabolism, but if they're allowed to accumulate, they can have a devastating effect on cells. The cytoskeleton, it's an elaborate network of protein structures that act as the cell's bones and muscles. Elements include microfilaments, intermediate filaments, and microtubules, which we'll talk about more on the next slide. Centrioles, paired, are called centrosomes. They lie close to the nucleus. They're rod-shaped bodies that lie at right angles to each other, and they generate microtubules and direct the formation of mitotic spindle during cell division, which we will learn more about later on in the lecture. Before I go into the slide, I just want to mention that table 3.1, the parts of the cell structure and function in your book, is stellar. It tells you the cell part, the structure, and the function in pretty good detail. There are, as you can see, many, many parts, but I like the way that it breaks down, structure, anatomy, and function for you, physiology. So take a look at that when you get a chance. So these are the elements of the cytoskeleton that we just outlined on the last slide, but they support the cell and help to generate movement. So microfilaments are most involved in cell motility and producing changes in shape. Intermediate filaments are strong, stable, and rope-like, made up of fibrous subunits, and they help form desmosomes, which we learned about as a cell membrane junction, and provide internal guy wires to resist pulling forces of the cell. And then tube-like microtubules are made up of repeating subunits of the protein tubulin. They determine overall shape of the cell and the distribution of the organelles, and they're important in cell division. Now let's talk about cell extensions. So first we have cilia and flagella, which sound like the names of the evil stepsisters in Cinderella or something, but they kind of look like eyelashes. So cilia are whip-like cellular extensions that move substances along the cell surface. So this would be an example of how mucus is able to leave the lungs. And when a cell is about to make cilia, its centrioles multiply and then line up beneath the plasma membrane at the free cell surface. And then microtubules then begin to sprout from the centrioles and put pressure on the membrane, forming these projections. If the projected form of the centrioles are substantially longer, they're called flagella. So flagella, like I just said, they're longer and they propel a cell itself. So an example of this would be sperm. And then microvilli, little shaggy hairs, tiny finger-like extensions of the plasma membrane that project from an exposed cell surface, and they increase the surface area. Usually found on the surface of cells active in absorption, such as intestinal and kidney tubule cells. And the reason why is just what I said, that they increase surface area and therefore increase that area that is available for absorption. Up until this point, we've been learning about the different parts of the cell and about the cell as a generalized cell. However, there is a lot of cell diversity in the human body. There's trillions of cells with about 200 different functions. They range in size from 1/12,000th of an inch to 3 feet. Nerve cells that enable you to wiggle your toes. So there's a lot of variability, and a lot of the amount of organelles they have will determine function. Their shape determines function, which we've kind of alluded to when we're talking about things like mitochondria. The tightness of epithelial cells, that's something that would line like the inside of your cheek and create this living barrier so that bacteria can't easily enter your body. So here are some different cell types in the human body. So cells that connect body parts are called fibroblasts and erythrocytes, which are red blood cells. Cells that cover and line body organs are epithelial cells. Cells that move organs and body parts are skeletal, cardiac, and smooth muscle cells. Cells that store nutrients are fat cells. Cells that fight disease are white blood cells. And cells that gather information and control body function are nerve cells or neurons. And cells that are charged with reproduction, we have the oocyte in the female or sperm in the male. Now let's talk about membrane transport. So first we have to understand what a solution is. So a solution is a homogeneous mixture of 2 or more components, and the substance present in the largest amount is a solvent. So for example, water is the chief solvent in your body, and components present in smaller amounts are solutes. So intracellular fluid is a solution containing a small amount of gases, oxygen and carbon dioxide, nutrients and salts dissolved in water. And then extracellular fluid, aka interstitial fluid, continuously bathes the exterior of your cells. It has thousands of ingredients. The plasma membrane is selectively permeable. So selective permeability means that the barrier allows some substances to pass through while excluding others. So now let's talk about 2 ways substances can move across the membrane, via active transport or passive transport. First, let's talk about one of the passive processes, or one of the passive modes of transport, diffusion. So diffusion is movement away from areas of higher concentration or just movement down a concentration gradient. So you can see here in the image below the definition, it's a beaker, you put in a tablet that dissolves, and pretty soon that dissolved tablet will be equally distributed throughout the whole beaker of water. Another example similar to this would be like a tea bag in a hot glass of water. So there's simple diffusion, which is just unassisted, osmosis, which is water diffusion, and then facilitated diffusion. So something is helping it along. A membrane protein channel or a membrane protein acts as a carrier. So the hydrophobic core of the plasma membrane is a physical barrier to diffusion. However, they will diffuse if any, or allow diffusion if any of the following are true. So molecules are small enough to pass through the pores. So like in osmosis, the water diffusion via aquaporin. The items are lipid soluble, or it's assisted by a membrane carrier. So one of those membrane protein channels or a membrane protein acting as an actual carrier. So this image is just showing the different types of diffusion. So again, a simple diffusion, the lipid soluble solute directly goes through the phospholipid bilayer. So it's lipid soluble. There's more lipid soluble solutes on the outside in the extracellular fluid, so it moves down the concentration gradient into the cytoplasm. Osmosis, diffusion of water through a specific channel protein. You can see that aquaporin, and those water molecules are just able to go right on through. Facilitated, so through a protein channel, or through a channel protein rather, mostly ions selected on a basis of size and charge. And then the facilitated diffusion. You can see in the first part of image D that the lipid insoluble solutes kind of fit into that protein channel, and the carrier protein, it binds to it, and then it just opens up on the other side and allows it into the cell. Another passive process is filtration. So this is just the process by which water and solutes are forced through the membrane by fluid pressure, which is also known as hydrostatic pressure. It moves down the pressure gradient, or moves because of a pressure gradient. So in the kidneys, water and small solutes filter out of the capillaries into the kidney tubules because the blood pressure in the capillaries is greater than the fluid pressure in the tubules. It is not selective. Now let's talk about some active processes of transport. So this is sometimes called solute pumping, but active transport is quite similar to facilitated diffusion in that both processes require protein carriers that interact specifically and reversibly with the substances to be transported across the membrane. However, facilitated diffusion is driven by kinetic energy of the diffusing molecules, whereas active transport actually utilizes ATP to energize those protein carriers. And those protein carriers are called solute pumps. So basically, just like I said, when the cell has to use ATP to move those substances across the membrane, it's an active process. It's useful for items that are too large to pass through the membrane channels, or the membrane may lack the special protein carriers for their transport, or they may not be able to dissolve in the fat core, or they may have to move uphill against a concentration gradient. So there's active transport and vesicular transport. We've already heard the word vesicular a few times when we were talking about those organelles. So again, solute pumps utilize ATP. Active transport, used for amino acids, some sugars, and most ions. And here on the right, you can see the sodium-potassium pump. It alternately carries sodium ions out of and potassium ions into the cell. This process is absolutely necessary for normal transmission of nerve pulses. So this is a very important function. There are more sodium ions outside of the cell than inside, so those inside tend to remain in the cell unless the cell uses ATP to force or pump them out. ATP is split into ADP and one inorganic phosphate, and the phosphate is then attached to the sodium-potassium pump in the process called phosphorylation. Likewise, there are more potassium ions inside cells than in the extracellular fluid, and the potassium ions that leak out of the cells must be actively pumped back inside. Because each of the pumps in the plasma membrane transports only specific substances, active transport provides a way for the cell to be very selective in cases where substances cannot pass by diffusion. No pump, no transport. This image is kind of cool. So you can actually see that inorganic phosphate is kind of acting like a key to initiate that pump when it comes off the ATP and turns into ADP. Now let's talk about the other active process of transport, vesicular transport. So we have exocytosis and endocytosis. Exo would be like exiting out of the cell. It's how the secretion of hormones, mucus, and other cellular products occur. Ejection of cellular waste occur this way. Packaged into a vesicle, migrates and fuses with that plasma membrane and ruptures, filling its contents outside of the cell. Endocytosis into the cell engulfs extracellular substances by enclosing them in a vesicle, and just does the opposite, detaches from the plasma membrane and moves into the cytoplasm. In some cases, travels to the other side and expels content by exocytosis there. So it does involve ATP to fuse and separate membrane vesicles and the cell membrane, and it moves substances in and out of the cells in bulk. The image on the left is demonstrating exocytosis. Again, the mechanism that cells use to actively secrete hormones, mucus, and other cellular products, or to eject certain cellular waste. The product must be released. The product to be released must first be packaged in that vesicle. It typically occurs via the Golgi apparatus into a secretory vesicle. So, a secretory vesicle because it's going to secrete. The vesicle migrates to the plasma membrane, fuses with it, and then ruptures, filling the contents. You can also see that in those pathways we were talking about from the Golgi apparatus earlier in the lecture. That was pathway 1, where it just secretes the contents into the extracellular fluid. It involves a docking process in which docking proteins on the vesicles recognize that plasma membrane via its docking proteins and binds with them. And that binding causes the membranes to quote-unquote corkscrew together. So it basically goes into its little secretory vesicle and migrates to the plasma membrane, screws on, and opens up and releases all of that, whatever it is, into the extracellular fluid. And on the right, we have endocytosis, an image demonstrating endocytosis. And you can see the sequence of events is a vesicle forms by forming a pit in the membrane. And once that vesicle detaches from the plasma membrane, its contents may be digested by a lysosome and then released to the cytosol, or the vesicle may be transported across the cell intact and then released to the cell exterior by exocytosis. So it might just be traveling through. If present, its membrane components and receptors are recycled to the plasma membrane. And the type illustrated is pinocytosis, phagocytosis, and receptor-mediated endocytosis. So, if the engulfed substances are relatively large, things like bacteria, dead body cells, and the cell separates them from the external environment, the endocytosis process is more specifically called phagocytosis. So, that's part of the immunological system. So we've mentioned it a couple times when we're talking about white blood cells. But so you can bet that anything that has to do with bacteria or dead body cells, things that need to exit the body, things that need to go away, the process by which that will happen is phagocytosis. Penocytosis is cell drinking, quote unquote, during which the cell gulps droplets of extracellular fluid. The book example to help you kind of remember what penocytosis means. Just imagine somebody drinking Pinot Noir. The plasma membrane indents to form a tiny pit or cup to hold your Pinot Noir, and then its edges fuse around the droplet of extracellular fluid containing dissolved proteins or fats. So unlike phagocytosis, pinocytosis is routine activity for most cells. It's especially important in cells that function in absorption, so like those in the lining of the small intestine. And then receptor-mediated endocytosis. It's the main cellular mechanism for taking out specific target molecules. So receptor proteins on the plasma membrane bind exclusively with certain substances. Both the receptors and high concentrations of attached target molecules are internalized in a vesicle, and then the contents of the vesicle are dealt with in one of the ways shown in this figure. All the phagocytosis and pinocytosis are important. They are not very selective compared to receptor-mediated endocytosis. Specific substances taken in via this route include things like enzymes, hormones, cholesterol, iron. And one of the hiccups of the immunological system is that flu viruses will exploit this route to enter and attack cells. Now let's get into the cell life cycle. So there are two stages of the life cycle. The first is called interphase. So where the cell grows and carries on, it's living its best life and just doing its usual metabolic activities. And then cell division. Everything else is cell division. Cell reproduces itself. Its function is to produce more cells with the same genetic material, and DNA replication occurs before cell division. So interphase is everything that's not cell division. So let's talk about mitosis. So it's the process of dividing that nucleus into two daughter nuclei. As I mentioned on the previous slide, DNA has already replicated before this begins. So this single cell will have double the genes for this period of time. So the image you can see interphase is there at the beginning. So interphase is everything before this all goes into motion, before all the cell division begins. And then we have prophase. So as the cell division begins, the chromatin threads coil and shorten so that the bar-like chromosomes become visible under a microscope. Because DNA has already been replicated, each chromosome is usually made up of two identical strands called sister chromatids, held together by a small button-like body called a centromere. The centrioles separate from each other and begin to move towards opposite sides of the cell, directing the assembly of a mitotic spindle. It's composed of a microtubule. Between them as they move. The spindle provides scaffolding for attachment and movement of the chromosomes during the later mitotic stages. And by the end of prophase, the nuclear envelope and the nuclei have broken down and temporarily disappeared. And the chromosomes have attached randomly to the spindle fibers by their centromeres. And then we enter metaphase. In this short stage, the chromosomes line up at the metaphase plate at the center of the spindle midway between the centrioles, so that a straight line of chromosomes is seen. So you can see in the bottom left of the figure that all of them are lining up right along the center, along the metaphase plate. Next is anaphase. During anaphase, the centromeres that have held the chromatids together split, and the chromatids, now called chromosomes again, begin to move slowly apart, drawn towards opposite ends of the cell. The chromosomes seem to be pulled by their half centromeres, with their arms dangling behind them. So you can kind of see that in that middle bottom picture. This careful division of sister chromatids ensures that each daughter cell gets one copy of every chromosome. Anaphase is over when the chromosomes stop moving. Telophase is essentially prophase in reverse. Chromosomes at the opposite ends of the cells uncoil and become thread-like chromatin again. The spindle breaks down and disappears, and a nuclear envelope forms around each chromatin mass, and nuclei appear in each daughter nuclei. Nucleoli, appear in each daughter nuclei. Mitosis is basically the same in all animal cells, and depending on the type of tissue, it takes 5 minutes to several hours to complete. You can see here, after that nuclear envelope forms, after telophase, we have cytokinesis, which is just the division of the two cells, or the one cell into two. It's the division of the cytoplasm. It usually begins during late anaphase and completes in telophase. So that cleavage furrow forms. It's made up of microfilaments over that midline and just squeezes or pinches the original cytoplasmic mass into two parts. So mitosis and cytokinesis go hand in hand, but in some cases, the cytoplasm is not divided. This condition leads to the formation of binucleate, or two nuclei, or multinucleate cells. It's fairly common in things like the liver and the skeletal system. So it's not abnormal for the cytoplasm to not divide and to just leave a cell with multiple nuclei. I want to briefly talk about protein synthesis. So we know that proteins are key substances for all aspects of life. They're major building materials for the cells. They perform functional roles in the body, like enzymes, which are very important. So obviously cells need to know how to, it is important that they produce proteins. So that's called protein synthesis. Protein synthesis occurs via different modalities. So first we have transcription. So that's when complementary mRNA, the messenger RNA, is made using the formation of a DNA gene. In translation, when the information carried in mRNA molecules is decoded and translated from nucleic acids into proteins. So that's what you can see happening in this figure. Step 1, transcription, all the way to translation. So step 3 ensures that the correct amino acid is added at the correct location in the growing protein. So a little more on transcription. This is the process of converting oral conversation to a written record in human world. So that language applies to proteins. It's just converting that information into the same language. But in this context, the language is the nucleic acids. So transcription involves the transfer of information from the sequence of bases in a DNA gene into the complementary sequence of mRNA by an enzyme. DNA is the template for the transcription and mRNA is the product. Each 3-base sequence specifying a particular amino acid in the DNA gene is called a triplet. And the corresponding 3-base sequence on mRNA are called codons. So translation is the phase of protein synthesis where the language of the nucleic acids, that base sequence, is translated into the language of the proteins, so the amino acid sequence. And translation occurs in the cytoplasm and involves all 3 major varieties of RNA. Translation consists of the following series of events, which is illustrated in steps 2 through 5. So the mRNA attaches to the ribosome, tRNA transfers or delivers amino acids to the ribosome where they are linked together by peptide bonds formed by dehydration synthesis. So the water is taken away in the exact sequence specified by the gene and its mRNA. There are about 45 common types of tRNAs, each capable of carrying one of the 20 types of amino acids. And that is not the only job of tRNAs. They also have to recognize the mRNA codons to double check that the amino acid they are coding will be added in the correct order. They can do this because they have a special 3-base sequence called an anticodon on their head that can temporarily bind to the complementary codons. Once the tRNA delivers its amino acid in the correct position at the beginning of the mRNA message, the ribosome moves to the mRNA strand along, bringing the next codon into position to be read by another tRNA. As amino acids are brought to their proper positions along the length of the mRNA, they are joined together with peptide bonds catalyzed by a large ribosomal subunit and begin folding into that final protein structure. As each amino acid is added to the chain, its tRNA is released and moves away from the ribosome to pick up another amino acid. When the last codon, the termination or stop codon, is read, the protein is released. So that's step 5. Let's move on to part 2 of chapter 3. We've talked quite a bit now about cells, but when you group cells together that are similar in structure and function, you get tissues. And then tissues further on are organized into organs. There are 4 types of tissues, so epithelial, connective, muscle, and nervous. So epithelial tissues are the covering tissues. Connective tissues support the body. muscle tissues are charged with movement, and nervous tissues are charged with control. Epithelial tissue. So also known as the epithelium, it's the lining, covering, and glandular tissue of the body. It covers all free body surfaces, both inside and out, and contains versatile cells. So the functions are protection, absorption, filtration, and secretion. So the skin covering your entire body is epithelial tissue. If you recall, we did talk about how epithelial cells fit closely together. They're packed in tight so that they provide that protective barrier for you. Apical surfaces is when one free, unattached edge is exposed to the body's exterior or to the cavity of an internal organ, and it's slick and smooth, or it may have microvilli or cilia. Basement membrane is where the anchored or basal epithelium rests, and it's a structureless material secreted by both the epithelial cells and the connective tissue cells deep in the epithelium. It is avascular. When well nourished, it can regenerate itself. So by avascular, I mean there's no direct blood supply to it. There is a language to classifying epithelia. So we have simple and stratified, and this is just based on the number of cell layers. So simple being single layer, stratifying being multiple. Then we have squamous, cuboidal, and columnar, and this is classification based on cell shape. So if you have single layer, so simple squamous, the function is diffusion and filtration and the secretion in serous membranes. If you have simple cuboidal or columnar, it's charged with secretion, absorption. Ciliated types repel mucus or reproductive cells. If you have stratified squamous, it's charged with protection. Transitional epithelium is modified. Stratified squamous stretches to accommodate distension of urinary structures. So you're going to see transitional epithelium in your bladder, for example. Cuboidal, columnar, stratified types offer protection, and these tissue types are rare in humans. There is a really good figure in chapter 3. It's figure 318, just shows you the types of epithelia and examples of common locations in the body. Aside from being important to know, it's also just very interesting because it shows you the microscopic versions of these as well. But just really quickly, simple squamous cells can be found in the alveolar walls of the lungs, so those air sacs in the lungs. Simple cuboidal can be found in the epithelium of kidney tubules. Simple columnar can be found in the epithelium of the small intestines. Pseudostratified and ciliated columnar can be found in the epithelial lining of the human trachea. And then stratified squamous can be found in the epithelium lining of the esophagus. The last photo in the figure that it shows is the transitional epithelium, which we just talked about. It's in the bladder. So in a relaxed state versus when the cells flatten and elongate when the bladder fills with urine. Definitely go check out that figure in your book. Now let's talk about connective tissue. Miraculously, the meaning is in the word. The meaning is in the name. It connects body parts. It's found everywhere in the body, and it's the most abundant and widely distributed of the tissue types. Performs many functions, primarily involved in protecting, supporting, cushioning, and insulating other body tissues. It is vascularized, and it utilizes an extracellular matrix. So the extracellular matrix is made up of different types of cells plus varying amounts of non-living substances found outside the cells. You can read more in your text about the extracellular matrix and how it makes connective tissues different from the other three types. So the types of connective tissues, we have bone, cartilage, dense connective tissue versus loose connective tissue, and then blood is actually considered a connective tissue. It's considered connective because it consists of blood cells surrounded by a non-living fluid matrix called blood plasma, and the fibers, quote unquote, of blood are soluble proteins that become visible only during blood clotting. Then we have muscle tissue. So you can see here there's 3 different types. We first have skeletal muscle. It's packaged by connective tissue sheets into organs, which are attached to the skeleton. It's voluntary, form the flesh of the body, aka the muscular system. When skeletal muscle contracts, it pulls on bones or skin. It's responsible for gross body movements, in other words, like your large body movements or changes in facial expression. It's long and cylindrical, as you can see in the image. It's multinucleate with obvious striations that are often called muscle fibers. And then we can move on to the right, cardiac muscle, which is found in the heart wall. I'm not going to go super far into this because we're going to have a whole cardiac chapter, but it does have striations. It's single nucleus, so one nucleus, and the cells are relatively short. It is an involuntary muscle. I want you to think about this for a second, why it's important that cardiac muscle is an involuntary muscle. So if we had to be actively and willfully make our heart beat, it would be a lot of work. So it's good that this isn't voluntary. It just does it for us. And then at the bottom we have smooth muscle. There's no striation visible. It's single nucleus and tapered at both ends. It's found in the walls of hollow organs such as the stomach, uterus, and blood vessels. As it contracts, the cavity of the organ alternately becomes smaller or enlarges, so substances are mixed and or propelled through the organ along a specific pathway. So an example of that would be peristalsis. So peristalsis is just the movement of foodstuffs and food waste through your intestines, through your GI system. Then we have nervous tissue. So the cells are called neurons. All neurons receive and send electrochemical impulses from one part of the body to another. So their major functions are irritability and conductivity. The structure is very unique. So that cytoplasm is drawn out into long processes, extensions as long as 3 feet or more. So that's how we're able to wiggle our toes or feel things in our extremities. The nervous system is more than just neurons. A special group of supporting cells called neuralgia insulate, support, and protect the delicate neurons in the nervous system, which is comprised of the brain, the spinal cord, and the nerves. Now let's talk about tissue repair. So when a wound occurs in the tissue, it can spark 1 and or 2 responses. So there'll be inflammation and then possibly an immune response. So inflammation is very general, it's non-specific. Immune response is going to be extremely specific. It's when your immune system recognizes foreign invaders like bacteria, viruses, etc. We'll talk about more in depth about that in chapter 12. Here, let's focus just on the actual repairing of the tissue, so wound healing. Ultimately, 2 things occur, regeneration and fibrosis. So regeneration is going to be the replacement of that destroyed tissue, and then fibrosis is going to involve more dense connective tissue that helps form a scar. So tissue injury occurs and inflammation sets the stage. So these injured cells and others release inflammatory chemicals that cause the capillaries to become very permeable. This allows fluid-rich in clotting proteins and other substances to seep into the injured area from the bloodstream. So this is like you skin your knee and you get that, after it's done bleeding and it's trying to heal, you get that thick goop. That's part of it. Everything's coming to that site to heal. The leaked clotting proteins construct a clot, which plug the hole to stop things like blood loss and hold the edges of the wound together. The injured area becomes walled off, preventing bacteria or other harmful substances from spreading to the surrounding tissues. And where the clot is exposed to air, it quickly dries or hardens, forming a scab. Then granulation tissue forms. So here's our little critical thinking points that to think about, bring some notes, we'll discuss it in class. So hydrocortisone is an anti-inflammatory drug that stabilizes lysosomal membranes. Explain how this effect reduces cell damage and inflammation. And then think carefully about the chemistry of the plasma membrane. And then answer this question. Why is minor damage to the membrane usually not a problem? So I alluded to this earlier in the lecture, but think about how a disruption in that cell plasma membrane can not very difficult for it to correct itself. Any questions, comments, or concerns? Feel free to send them my way, drop them in the class discussion board, or bring them with you to the Zoom meeting and we can discuss as a class. All right, thank you. See you Wednesday.